Hey guys, I'm going to be talking about the HIV infection pathway. Um, so basically, to start off, I have a drawing here. It's showing the pathway from the virus entering a host cell and leaving the host cell. So first off, in your first step, you're going to have the host cell, you're going to have the viral particle entering your cell, right? So the virus enters the host cell. Now, step two, we want the viral RNA genome to be converted to viral DNA. Now, how does this happen? So, the virus has this thing called reverse transcriptase, which is an enzyme that makes double-stranded DNA, a double-stranded DNA copy of the viral RNA genome, right? So, during this step, the viral reverse transcriptase. So that's an enzyme that's going to make your copy of double-stranded DNA from the viral RNA genome. And so from there, the DNA, the copied DNA, is going to enter the nucleus of the host cell. And so we want the DNA copy of the viral genome to enter the host cell DNA. But how does this happen? How does it actually combine / insert itself? So here, if I draw this out to the side, right, we need a thing called integrase, right? That's another enzyme that's responsible for splicing the DNA into the host cell DNA, right? The viral DNA into the host cell DNA. So the enzyme integrase it does its work here. And once the viral DNA is inserted into the host DNA in the nucleus, we call that the provirus, right? So after insertion of virus DNA, we call that the provirus, right? So at this point in time, the provirus is dormant. So it means it's being replicated and passed on as the cell divides. So, but in this state, when it's dormant, the virus is essentially hidden from your immune system. However, when the provirus ceases to be dormant, right? So it's no longer dormant. If we look down here, so when it ceases to be dormant, that means it's stimulated to grow and divide. Step 4, right? So your virus is going to be transcribed to produce the viral RNA genomes that once entered the cell, right? That were up here in step 2. So it's going to transcribe the viral RNA genomes and viral mRNAs, right? And the viral mRNAs are translated to make the viral proteins, which I show here on the right side. And if I circle this, right? So transcription, you get the viral mRNA, and then translation of the viral mRNA, you get the viral proteins. Now, let's see here. Hold on. Transcription occurs here when provirus ceases to be dormant. Excuse my handwriting. And so basically, in this step, you produce your viral RNA genome and your viral mRNA, which is then translated, and you get your viral proteins. Right? So in step 5, I'm going to draw it down here because I'm running out of space. Right? This is step 5. So in step 5, your viral RNAs are going to combine with your viral proteins, and they're going to create your new viral particles. So if I write it here, the viral RNA plus your viral proteins, you're going to be making new viral particles. Right? So how do these viral particles that were just made, how do they exit the cell? Well, they exit via budding. That's what I'm showing here. The plasma membrane is slowly budding off, so it exits via budding. Now, the HIV virus, it does have an envelope in its structure. The envelope comes from the host cell's plasma membrane, and it also has the embedded viral glycoproteins, which coat the particle during budding, right? So once this buds off, right, after budding, you get a free infectious HIV particle, right? And this new HIV particle that just now bud off from your host cell, it's going to be able to infect other cells, right? And so that right there is the HIV infection pathways pretty much simplified.